Okay, 2.1 cell structure. So we back, new year, new term, new stuff. Let's get it. In bio, we start almost every unit with the cell theory, which states three things. All living things are made up of one or more cells. All cells come from pre-existing cells through cellular division. The cell is the basic and functional unit of structure and function in living organisms. Pretty simple stuff. All right. The major types of cells are prokaryotic and eukaryotic. What the heck does it mean? Okay. So eukaryotes have nucleus, they have linear DNA, and they contain membrane organelles like mitochondria, chloroplast, and the nucleus. Prokaryotes doesn't have all of the above I just stated. Prokaryotes don't have nucleus, they don't have they have circular DNA, but they don't have linear DNA, and they don't have membrane organelles. So the five kingdoms are prokaryotes, protista, plantae, fungi, and animals. This is pretty weird, as you can see that prokaryotes include eubacteria and archaea. Bacteria and archaea. Archaea. They're pretty similar, but there are some differences. That's why they're given different names. But under the eukaryotes, there are protista, plants, fungi, animals. And we will go on to explore the differences between them. So prokaryotic and eukaryotic, what are the differences? The tip says to compare prokaryotic and eukaryotic cells with respect to their size, internal organization, shape, and location of chromosomes. We're going to say that eukaryotes are a lot larger in size, and internal organization-wise, eukaryotes have membrane-bound organelles. They have nucleus, mitochondria, ER, and chloroplast, and etc. But prokaryotes just have some water, cytoplasm, and DNA floating around inside its body. It's pretty boring, and it's tiny. You can't see it. Yeah, kind of lame. Shape and location of chromosomes. For prokaryotes, the we don't really call its DNA chromosomes. Usually for chromosomes in biology, we are referring to the X shape. We're referring to the very condensed DNA that are seen in during mitosis or like meiosis. In prokaryotes, DNA is not that condensed. They're just floating around in the cytosol. They're just floating around in water, like vegetables in a soup. But in eukaryotes, they can be found in condensed form, like X shape, or just a single strand. We call this linear DNA. And in eukaryotes, they are found in nucleus. Yeah. So blah blah blah. Yeah. Okay. All these things. ER, ER. ER stands for endoplasmic reticulum. Long name. Do you need to remember it? No. What does it do? So there are two types of ER: smooth ER, SER, left bottom, and rough ER on the right half. So what's the difference? They're both called ER. What's the difference? Well, rough ER has a thing called ribosomes attached to it. If you remember our topic from last term, we learned that ribosomes are what makes proteins. So ribosomes join up amino acids into amino acid chains, and they fold it into a specific structure that it becomes the protein and functions the way it does. So that's what ribosome does. And rough ER synthesizes proteins. Now you can guess why the ribosome is there, because without ribosomes, it's impossible to synthesize protein. But SER doesn't have the ribosome, and since it doesn't have it, it synthesizes fats and lipids, and all these products, when they're synthesized, they're sent off to the Golgi body. And that's what we have next, the Golgi body. The Golgi body receives vesicles that are sent from other parts of the other organelles, and they can make changes to it. They can fold it. They can fold it into a different structure, and they can send these products. They can pack these products in a small package, like a vesicle, and secrete it to other parts of the body. They can send it off to other parts of the body or just around the cell. That's why Golgi body is called the post office of the cell. And mitochondria, our favorite organelle. Mitochondria is the powerhouse of the cell, as we learned from year 8. So why is it called the powerhouse? Well, it synthesizes ATP, the most important molecule for the survival of anything, of living things. So mitochondria is just looks like a worm, looks like the bowl of not the bowl of virus. It looks like a thick worm, very thick. And inside it there are folded membranes. You can see this folded structure kind of looks like your large intestine, very folded. This is because more folding actually maximizes the surface area, and you want in a cell you want to have more surface area because that maximizes the efficiency of chemical reactions. A few things. The matrix is just referring to this empty space in mitochondria, while crista is pointing to this folded curve right here. Like that small curve is called a crista. And there are also ribosomes attached to the mitochondria. Why is that? Well, mitochondria needs enzymes to speed up the reaction. So sometimes it needs to synthesize that enzyme. Therefore, there's the ribosome to synthesize protein, the enzyme. Chloroplast. Now, chloroplast is only found in animals, not in in plants. My bad, plants, not animals. We don't need chloroplast. And the stroma is just referring to the liquid, the gel-like liquid that's in the chloroplast. That's the stroma. And a thylakoid is one of these small green pancake-like structure, just a single layer, whereas granum is an entire stack of thylakoids. Granum, one thylakoid, a stack granum. What do they do? Well, the chloroplast just captures light energy and makes sugar from it. And blah blah blah. Don't really need to see this. Cytoskeleton, they are interesting stuff that we didn't cover in year 11, but they're basically just structures that are present in the cell that is used to support the structure and used to sometimes transport things around the cell. Okay, plant and animal cells, cell membrane, fluid mosaic model. Again, this was something we covered last year, but doesn't hurt to go over it again. So when we're talking about membrane, we usually picture it as a sheet of paper, but thinner. But in real life, when you zoom into a cell membrane, it's actually a lot complicated. There are proteins all over this membrane, and we call those ones on the surface the peripheral. Where's peripheral protein? As you can see, that's just on the surface, and so is that's just on the surface. We call these ones peripheral protein. And for the ones that goes throughout the width of the membrane, we call it channel protein. Usually, channel proteins are used for, ignore what I said. For the ones that go throughout the width, we call them integral protein. Usually, integral proteins can be used to transport materials in and out the membrane. And an example of that is channel protein. But channel protein is a type of integral protein. So don't get these two confused. They can be confusing, and I just made the mistake myself. But that's all right. Let's go. Let's move on. Phospholipid. We know from year 8, year 9, year 10, year 11 that phospholipids have a hydrophilic head and hydrophobic tails. So the head is polar, which is which means that it's water absorbing because water is also a polar molecule. And so if you have water on this end of the membrane, the head would absorb it, but when it's in the middle of the membrane, it's going to be stopped, kind of stopped by the nonpolar tails. So nonpolar doesn't like polar, therefore the tails are hydrophobic. They don't like water at all. So when there's water present in the middle of the cell, in the middle of the cell membrane, it's going to move very, very slowly through to the other end. There's diffusion. Water can diffuse through this membrane, but it's going to do that very, very slowly because of the presence of the nonpolar tails. That's why we have these integral proteins, such as channel protein, some proteins that let the water through quicker than simple diffusion would. And there are 4 rows of membrane proteins. The one we just talked about, transport, second, receptors, 3, enzymes, for reaction at the membrane or within it, 4, cell recognition. And glycoproteins, markers recognized by membrane proteins on other cells. That's all.